Fries, for that wonderful talk. We will now be moving on to the Q&A portion. Let me get the screen shared. Okay, so if you have a question for Dr. Fries, please use the Q&A box, not the chat feature, to ask your question. We have several questions already in. The first question, it seems that it's the intrinsic and circuit properties of target regions, V4 in this case, that determine the best coherence band for optimal communication. Are these modulated, changed by other top-down or neuromodulator systems, like layer 1 VIP activity or acetylcholine modulation?
Thank you. That's a very interesting question. As I showed you, the gamma frequency is certainly changed by attentional top-down. So attentional top-down enhances the gamma frequency. We do not know exactly yet how it does this, but as you mentioned, there is evidence from the lab of Yam Dunn and partly from Mike Stryker that top-down input, at least in the mouse, acts via VIP neurons, which then might be activated. They might inhibit some neurons and thereby disinhibit the V1 V4 circuit, which actually generates the gamma, right? So this is one possibility. Of course, this is currently extremely hard to test in the monkey because we are lacking the genetic methods that can be used in the mouse.
Thank you. Another question from that same attendee. How do these rules translate for communication between higher order areas, between, say, V4 and other associative areas?
The question started, how do they translate?
Correct. How do they translate for communication between higher order areas?
Okay, so generally what we see is that in the supragranular layers that are more prominent in sending feed-forward projections, we see more gamma band rhythmicity. And in the feedback projecting layers, we see more alpha-beta activity. Generally, we and others have found these rhythms in very many areas, including higher level areas. So gamma band activity has, for example, also we have clearly seen it, for example, in TEO. Bijan Pesaran has shown it beautifully in lateral interparietal area. It has been shown in MT. We have found it in MT, has been found by Bob Desimone and others in frontal eye field. So it seems like this, and of course there is famous work from Yuri Buzaki in the hippocampus. So it seems like, and it's clearly present in the motor system, right, in the motor cortex. So it seems that gamma band activity is particularly strong in V1, but you can see it probably everywhere in the cortex where you study it appropriately. But there is probably a gradient where it is stronger in V1. V1 might be particularly equipped to have it very strongly, and higher areas might be particularly equipped to have stronger alpha-beta rhythms, which, as I showed, seem to convey top-down. So this seems to make sense. But we see gamma in higher areas, and we can also see beta in lower areas.
Perfect, thank you. Our next question, it looks like supragranular stimulation induces earlier and sharper responses than infragranular stimulation. Any guess why?
That's a very acute observation. I don't know. As I mentioned, this was a preliminary result in this cat experiment. We are now, as I showed, moving this to the awake macaque, and we'll do this, of course, systematically in many penetrations in a few animals. And I think then we can address this more. At the moment, I wouldn't want to speculate about this.
Okay, thank you. Our next question from Jonathan Baker. Do you think the optogenetic induced gamma in cat striate is similar to an arousal input? For example, electrical MRF stimulation as shown in 1996 and 2004 by Singer and colleagues.
Absolutely. So you are referring to the work of Matthias Munk when he was in the Singer lab. That was at the time when I was also a PhD student there. So, well, so the gamma induction itself That is not due to the mesencephalic reticular MRF stimulation, but gamma is induced simply by visual stimulation, or when we drive the circuit optogenetically, then we just see gamma. MRF is a modulating system, or maybe under anesthesia it could be enabling, but it is not so. The picture that I think I would draw is that the cortical circuit, at least in the awake state, is very prone, at least in visual cortex, to generate gamma. If you excited, there is sufficient excitatory drive, then the tight interaction between the excitatory neurons with probably the parvalbumin positive interneurons, this feedback circuit simply generates gamma. And what's specific about the cortex, right, is this tight interconnectivity between excitatory and inhibitory neurons. It is different in thalamic circuits, right? But in the cortex, that's how it is. And that probably generates a gamma resonance or beta resonance in deeper layers, maybe because the interneurons there are equipped with different properties, as for example, suggested by the work of Nancy Coppell. And then gamma is in some sense a very simple rhythm, right? Any excitatory drive, at least to the awake cortex, is generating this, be it sensory, like visual, or be it optogenetic. And actually with optogenetic drive, we've seen gamma that has been stronger than any gamma we've ever seen with visual drive. These other things are modulatory, right? Like attention is modulatory, MRF stimulation is modulatory.
Okay. Next question. Can you please provide the company again for the custom-made guide tubes?
Yes. This company is called Unimed. I can write it into the chat. Unimed. And it is situated in Lausanne. And they are specialized for medical tubing. So they can basically take a tube and put a tip on it, or they can cut it open on the side, or they can take a larger tube and make it thinner. I mean, it's amazing what they can do. Yeah. And such a guide tube, there is some setup cost, which I don't know off the top of my head, but in the end, then the individual guide tube, if you buy 50 of them or so, is actually not expensive. It's a few euros.
Okay.
So we have very good interactions with them.
Great. Next question from Jonathan Fritz. One question from the early beautiful experiments of Bosman et al. In 2012 is whether there are distinct and fixed gamma rate signatures for different parts of V1, representing different parts of the visual field, or whether the key point is that there are distinct but relative, not fixed frequencies for different areas of attention in the visual field.
Ooh, Jonathan, can you just repeat this question slowly?
Yes, of course. So one question from the early beautiful experiments of Bosman et al. In 2012 is whether there are distinct and fixed gamma rate signatures for different parts of V1, representing different parts of the visual field, or whether the key point is that there are distinct but relative, not fixed, frequencies for different areas of attention in the visual field.
Okay, so we use two stimuli that were of equal eccentricity, equal size, and equal contrast. In this case, you typically find visually induced gamma rhythms of the same overall frequency. So these two stimuli induce two local gamma rhythms at two distinct populations that have the same overall frequency typically, if attention is not considered for the moment. But they are not coherent with each other. Even if the two stimuli are present simultaneously, there is an absence of coherence between them. If then attention is directed to one of the two stimuli, then the corresponding gamma rhythm quite consistently increases the gamma frequency by a few hertz. And we find this interesting because other manipulations that enhance the salience of a stimulus also enhance the gamma frequency. For example, higher contrast, as shown by the lab of Mounsell first, and we found the same thing then also. Higher contrast leads to higher gamma frequency. Moving stimulation versus stationary leads to higher gamma frequency. being closer to the fovea leads to higher gamma frequency. Stimulus onset versus sustained stimulation leads to higher gamma frequency. So anything that seems to make the stimulus more salient, bottom-up salient or top-down attended, seems to enhance the gamma frequency. And we believe that this might be a kind of common currency of bottom-up or top-down salience, which is helping the more salient gamma rhythm to win the competition against lower frequency gamma rhythms. So Nancy Coppell tells me that if you have multiple oscillators driving a target oscillator, then the one with the highest gamma frequency is likely to win that basically dynamical systems competition.
Perfect. Thank you. The next question, when you penetrate with V probes, are you able to reuse the probes many times?
Yes.
Great.
I cannot tell you, actually I've seen that Elena is here on the call. I cannot tell you how often she is individually reusing a given, Elena, I don't know whether you can speak up. I see that you are in the participant list. So she is actually doing that. She might be able to tell you or write into the chat how often she has maximally reused a given vPro, but definitely they are reused.
Yes, if Elena, if you write it into the chat, we can broadcast that. But we can move on to the next question in the meantime. Is it surprising to see optosimulation of cat superficial layers only cause local excitation and no increased activity in deep layers? Why do you think this is the case? And do you confirm these electrodes are aligned parallel to the cortical column and have similar receptive fields throughout?
Well, I mean, so let's see how this is in the awake primate. Here in the anesthetized cat, it was such that the local injection of light into the supragranular layers caused neuronal activity, multi-unit activity, and gamma only there. It's of course a good question how surprising this is. I might be lacking some other studies that have done similar things. I'm not so much aware of it. Of course, people have studied, like, for example, Ed Callaway has studied what happens if you uncage glutamate in particular layers and then see what an individual neuron does. But to the best of my knowledge, and of course, electrical stimulation experiments have been done. To the best of my knowledge, I think not so much has been done in terms of optogenetically stimulating neurons specifically in different layers. So I didn't have a very strong expectation there. I guess my tendency was to expect that stimulating anywhere in such an intact system would activate the entire column, but that's not what we saw. And it could potentially have something to do with the fact that this was under anesthesia. So I'm very curious to see what will happen when we do this then soon in the awake macaque monkey.
The next question, why is the expression of the corticotropin releasing hormone receptor not present in the granular layers?
That's just phenomenological, but I think so. So the CAM kinase 2 alpha. So if we use, and others have reported the same thing, if one uses the CAM kinase 2 alpha promoter, then the expression is very strong. You can see exactly how it is in the bioRxiv paper, where we have respective histological sections. It's very strong in the superficial layers. It is present, but a bit weaker in the deep layers. And in the granular layers, you see fibers, but hardly any cell bodies. Honestly, I cannot tell you why that is. I'm not sure whether the granular layer has transcriptomically an absence of CAM kinase 2 alpha expression. It would actually be surprising to me because CAM kinase 2 alpha is generally meant to be a general promoter for excitatory neurons. I have to say that I'm not really an expert in this, but phenomenologically, this has been very consistent. We have done these experiments really in a substantial number of cats, and we have consistently found this. And as much as I remember, similar findings have been made by other published studies.
Okay. Next question. Do you implant ECOG arrays below dura, and have you compared epidural and below dura responses?
Yes, so in the awake macaque monkey, we have implanted them subdurally. I know that the lab of Andreas Kreiter has implanted them epidurally. And from him, I take that epidurally, he can also get defined receptor fields, but an overall signal to noise ratio that is substantially lower than if you put them subdurally. Now, I have to say that if you put them subdurally, you can record for a certain time, but at some point, the dura is encapsulating typically the ECOG, right? Because these are ECOG strips, and then the dura kind of grabs them. So with the time, the recording quality can decline somewhat. But overall, I think that the best way to do this is to implant them subdurally.
Dr. Fries, I just wanted to interject. So we still have a few questions left. We're technically past time, but if you want to stay to continue asking questions, we can do that, or we can send those questions to you after the webinar.
I'm perfectly fine if people want to stay on it, and I can try to be a bit faster in answering.
I think we still have good attendance and still a few questions, so I think the pace is fine, and we'll just continue.
It's fine with me.
Okay, perfect. The next question. Does the optimal suboptimal phase relationship in case of the V1 MT communication go hand in hand with changes in the strength of phase coupling? And in terms of analysis, how is it possible to discriminate the phase offset coupling strength effects?
So this pertains obviously to the analysis of the interareal phase relation that I showed at the beginning of the talk by Gustavo Rohnkohl. Just a little correction, this does not pertain to V1MT. I think that wasn't the question, but it's V1V4 in the awake macaque. So basically the question was how this phase relation relates to the coupling strength, if I recall correctly. Well, the coupling strength is typically quantified by the metric of coherence or phase locking value, and that measures how consistent the phase relation is at a particular value. So if on every single trial the phase relation is identical, then the phase locking value would be 1. And if it's completely random, then the phase locking value would be 0. So we basically use the fact that the phase relation on a trial-by-trial basis is not perfectly regular. So V1 gamma entrains before gamma at a typical phase relation. The fact that there is a mean phase relation and that it's not random shows that there is coherence, that there is some synchronization. And we then ask the question, the phase relation at which this happens, at which V1 typically entrains V4, is that optimal? Does that lead to the shortest reaction times as our metric of efficient communication? But we cannot basically, maybe that's what the question was meant to ask, on the trials, when the phase relation is close to the typical phase relation, we cannot, for that trial, basically, talk about the strength of phase locking. You could, of course, just say that if the phase relation on a given trial is exactly at the average phase relation, basically this trial is perfectly synchronized, if you will. It doesn't deviate at all from the average phase relation. And if it's 180 degrees to this, it would be maximally non-phase synchronized. That could be one way of talking about it.
Okay, great. The next question comes from the chat. My question is for beginners. Which probe would you recommend for optogenetic stimulations, and how many channels are minimum for reasonable recordings?
Okay, so we have the best. So I guess this now pertains to the laminar probes from Plexon. We have clearly the best experience with V probes. We have also tried S probes. In our hands, the unit recordings are particularly crisp with V probes. I think generally the S probes have been recommended for penetrating the dura. I showed you that at least with this approach that has been developed in my lab by Iris and by Elena and by Rasmus, we can use a blunt guide tube and we can reliably penetrate the dura, even though we are typically not scraping the dura. And then the V probes give us the best neuronal recordings. We also like them because at the tip they have the smallest dimensions. So of course with these probes there's always a bit of a cost that you pay with each penetration. There's a tiny little damage. So the smaller the damage, the better. And so the V probes are basically the slimmest at the tip there. They have the smallest footprint. I think the other question was how many channels? Well, so generally, I mean, the Hui one is surprisingly thin. So if you span about a bit more than 1.5 millimeters and you carefully position the electrode, you can have a complete depth recording. Higher areas are a little bit thicker. And then there's basically just a trade-off. You know, if you want to span that depth, then the fewer contacts you choose, the thinner your probe can be. Actually, you should know that we learned this recently, a few months ago, that there are standard dimensions on the Plexon website, right? So on the Plexon website, it says something like for, I don't know, 24 channels or so, it might be 236 micron diameter. And there's some sort of a standard price, but you can get them also thinner, smaller diameter for a somewhat higher price that at least at the time was not on the website. So that's what we are using. So we generally try to have the smallest footprint possible. And then there's just this trade-off, right? And generally we found that 236 micron diameter is okay for several penetrations, but generally the thinner the better. And in terms of the maximal spacing, it seems like 150 micron is okay to get a decent cross-spectral density, but of course, the denser, the better.
Okay, great. Next question. How would you explain that optogenetic stimulation in infragranular layers induce gamma?
That's a very good question. That's a bit of a surprise. I mean, in the awake macaque monkey, at least when we look at spike field coherence, right, in V1 and V2 and V4, this is the work by Beth Buffalo together with me and Bob Lesmer. There is a very clear effect that the deep layers primarily show beta. Now in the nest, so now if you look at local field potentials in deep layers, you can clearly also see visually induced gamma. For example, there's work by the lab of Bob Shapley and other work showing this. And what I showed you was actually local field potential. As I said, this was preliminary result. Also, this is an acetized cat. So I think for the moment, I wouldn't want to speculate too much about it. I think we need to investigate that in a system where there is beta. So if I have this correctly in my head, you know, beta might be related to SOM neurons, somatostatin positive interneurons. And if I recall this correctly, SOM neurons are very strongly affected by anesthesia. So they are almost silenced, I think. So I could imagine that this basically takes away much of the beta generation machinery and might give way to gamma if we drive the local circuit optogenetically. But honestly, this is really now going out on speculation. This is really, and I so take this with a big grain of salt. This is very preliminary results, and I think we need to do these experiments in the awake macaque.
Okay, I think this is our last question. How does optogenetics inform theta oscillations, e.g. Spropoulos PNAS 2018 study? Did you see that optogenetics induces theta in deep layers?
We haven't looked at this, so I cannot say. We have seen, you know, I told you that in the anesthetized animal there is hardly any alpha beta. Nevertheless, if you optogenetically stimulate, not only goes gamma up, but there's also a bit of a reduction in alpha, and maybe this goes a bit to theta. You should look this up in the bioRxiv paper by Christopher Lewis and Jianwang Ni and myself and others. But I cannot really tell you about theta in the anesthetized animal.
Okay, thank you so much again, Dr. Fries. We are all out of questions, and thank you to everyone else who attended. And make sure to catch our next segment of Advancing Research with Linear Probes next Wednesday at 10 a.m. Central. David Hersfeld of Duke University will be giving a talk entitled Using Multi-Contact Probes to Functionally Dissect Primate Floccular Circuit. So we'll see you next time. Thank you, everyone.
Thank you very much.